In Chapter 19, we are now going to bring all of the processes together that we have been discussing over the past few weeks. All of the oxidation of the sugars, fats, and amino acids we have discussed are going to converge into this final stage of cellular respiration. This process is going to account for the majority of the ATP produced inside of the body. We're going to go through parts of it, describing exactly how electrons are transmitted. We're then going to discuss how the electron transfer is coupled to ATP production, and then we'll end with a little bit on how this process is regulated. Before we begin, let's just make sure we remember what we were working through all of those oxidative pathways to produce. We have been producing NADH and FADH2. We've been calling these our reduced electron carriers, and we're now going to see how we get those reduced carriers to give us energy. We are going to take the reduced carriers and move them through an electron-transport chain, ultimately producing energy. Shown here in this diagram, we are going to take our carriers, jump them through several proteins of the electron-transport chain. During this process, a proton gradient will be produced, which will allow us to couple ATP and phosphate together, producing ATP. Oxygen is going to be our ultimate electron acceptor in this process. We're going to focus on how this works with humans. To describe the production of ATP, we need to discuss the Chemiosmotic Theory. In this theory, we are going to have the flow of protons moving down an electrochemical gradient, giving us some work, some energy to produce the ATP that we're trying to get. We've seen ATP produced throughout our metabolic pathways in other ways, where we were taking some sort of molecule that was actually higher energy than ATP, and then that molecule, such as phosphoenol pyruvate, for example, is actually high energy and is capable of donating a phosphate to ADP directly. That was our substrate level phosphorylation. We're now going to do the oxidative phosphorylation. To get this to happen, we are going to be moving protons against their natural gradient. So we have to have some other energy to move those protons in the opposite direction. The chemiosmotic theory is focused around the presence of a membrane that is impermeable to ions. Now in humans, which is what we're going to focus on, this is going to be the inner membrane of the mitochondria. There are other ways that this can occur in bacteria. They use the plasma membrane. With photosynthesis, they use membranes within the chloroplast, but we're going to focus on what's happening in the mitochondria. As we go through this, you need to keep this statement, the second statement here, in mind. What we're going to see is a coupling of the downhill flow of electrons. Downhill means that this is going to be a process that is spontaneous. It has a negative delta G. It's a reaction that we want to happen. The initial reaction we will see is the transfer of an electron from NADH to some other electron acceptor. That's going to be a downhill process, a naturally occurring process. We're coupling the downhill flow of electrons with the uphill flow of protons. The uphill flow of protons means that we're pushing those protons against the natural gradient. We're going to be moving the protons from the matrix into the intermembrane space. Once we get our proton gradient established, then the protons will have a downhill flow. The downhill flow will be the movement of the protons from the intermembrane space where we've been putting them back down through the ATPase into the matrix. That's the driving force that gives us the ability to phosphorylate the ADP, ultimately producing ATP. Now those two statements I just said, they are a mouthful. There's a lot involved with those two statements. You could even say those two statements are an overview of what we're going through. If we look at how these two things work using a picture, which sometimes is a little bit more helpful, what we've got in this picture, the outer space that's in white, that's representing the intermembrane space. So we're assuming the whole picture is the mitochondria. Then this yellow line outlined in blue, that's the intermembrane. The orangey color, that's the matrix. So the first thing that we're going to see is the reduced electron carrier, such as NADH, is going to donate an electron to a protein. That electron movement is going to be the downhill flow of electrons. That will cause the uphill flow of protons, the pumping of protons out into this intermembrane space. Now out in this intermembrane space, we have a high concentration of protons, also a lower pH. That will matter later. This high proton gradient will ultimately allow the downhill flow of protons through here, the driving force of ATP production. To really make sure we understand what's going on with this ATP production, we need to make sure we all understand exactly what's going on within the mitochondria. I'm going to flip to a picture to describe what's written on this page. Within the mitochondria, you have several structures. Starting with the outside, the outer membrane of the mitochondria itself is a relatively porous membrane. It has lots of pores throughout that allow pretty much any metabolite to pass as they would like throughout that outer membrane. Directly inside of the outer membrane is a region called the intermembrane space. The intermembrane space is pretty much like the cytosol of any other cell, but what's key here is we're going to have a really high proton concentration. The high proton concentration is because of the pumping that we saw here a few minutes ago. So this is the intermembrane space where we have all the protons we've been pumping. Within that, we will now see what we call the inner membrane. The inner membrane is nothing like the outer membrane. The inner membrane is impermeable. It doesn't let anything just pass directly through. What can pass through the inner membrane are protons, which is important to this whole process because we're looking at how protons can cross the intermembrane, giving us the proton gradient that we're looking for. Within the inner membrane, we have the matrix. The matrix is really where most of the stuff happens. The matrix is where we've seen the citric acid cycle occur. We saw the lipid catabolism, the amino acid catabolism. We've talked about that so far. So several things are occurring within this matrix. The matrix is also going to be where the protons are pumping from as we discuss the electron-transport chain. Okay. So if the electron-transport chain is allowing electrons to move from one thing to another, we've got to have some proteins in there. And within those proteins, we have to have some molecules or some structures that allow the binding of electrons. And we call these electron carriers. There's three major types of electron carriers that we're going to see throughout this. We're going to see flavin mononucleotide and then the FAD, which we've seen before, that's going to interact with complex 1 and 2. We're going to see cytochromes, A, B, and C. And then we're going to see iron-sulfur clusters, as well as coenzyme Q or ubiquinone, which is not on this list. And we're going to look at each of these individually. As we go through these different types of electron carriers, we need to remember there are three types of electron transfers that can occur. We can have a direct electron transfer, which is something that could occur between something like iron 3 and it gets an electron and it becomes iron 2. Or copper can do the same thing. We may also see a transfer of a hydrogen atom, which would be a proton and an electron being transferred at the same time. Or sometimes we will see a transfer of a hydride, which is a proton with 2 electrons at the same time. So just picking a place to start, let's take a look at what cytochromes look like. There are different types of cytochromes, and the different cytochromes have some slight differences, but we want to look at what's similar among all cytochromes. All cytochromes are going to have a heme in the center. The heme, of course, has an iron in the center, and that iron in the center is what's going to be able to accept the electron or donate the electron and allow this to be an electron carrier. Cytochromes are capable of carrying 1 electron at a time. This is where we will see the iron in the +3 state going to the +2 state, or vice versa, depending on whether it's being oxidized or reduced. All of these cytochromes are typically going to, well, not all of them, the cytochrome C, which we'll see the most, is typically going to be bound by a cysteine residue of a protein. And recall back from our earlier discussions, when you have a cysteine residue bound, it's going to be a covalent attachment, typically through a disulfide bond of some type. Now we need to look at the iron-sulfur clusters. Again, these are going to be capable of carrying 1 electron at a time, and they're also going to be bound to cysteine residues within the proteins. These are not hemes. They are simply iron molecules that are clustered together with sulfur molecules. Now we take a look at coenzyme Q, or also known as ubiquinone. These are exactly the same thing. Just because of the way I was taught, I will most likely call this coenzyme Q as we move along, but you have to keep it straight in your mind that coenzyme Q is the same thing as ubiquinone. Coenzyme Q is a little bit different than the other electron carriers. It is actually capable of carrying either 1 or 2 electrons. It is also lipid soluble, and therefore it's capable of moving within the inner membrane of the mitochondria from one protein to another. These figures here show you what it looks like as the the coenzyme Q accepts an electron. If 1 electron is accepted, we end up with a radical, and that's going to be important through a process we'll see called the Q cycle, as well as possible oxidative damage within the cell. If this free radical accepts another electron, then it becomes what we call ubiquinol. It's abbreviated QH2. So we're going to see this QH2 fully reduced form of coenzyme Q quite a bit as we get moving along. Now we're not going to do any math in this section of the class, but what I wanted you to see is it actually can be calculated, the amount of potential that we have once we form this proton gradient. You can sit down and you can do the math and figure out how the movement of electrons can change the pH and can therefore change the electrochemical or the reduction potential. The easy way of saying this is we get some free energy released as we move the protons. We store that energy in the presence of the proton gradient, and then as we dissolve or get rid of the proton gradient as they flow back through, that gives us the energy we need to make ATP. So now we're ready. Let's start looking at how these electrons jump from one protein complex to another and hopefully get to the point where we can make some ATP. This first figure is just to show you how the electrons can enter the chain. And then we'll go through each protein one at a time. Electrons typically enter the electron-transport chain in the form of NADH or the form of FADH2, which is what we would expect as we've been going along. We will start a discussion with complex 1, which is this pink blob here. And you can see that NADH is going to interact here, donating an electron to complex 1, becomes NAD. And just to make sure you realize, this NAD is now oxidized. It can go back in any of the other reactions that we've seen except another electron and then come back to donate to complex 1 again. This reaction here should look familiar. Succinate becoming fumarate, that is part of the citric acid cycle that we saw, and this is the one that produced FADH2. So this reaction directly puts an electron into complex 2. We also saw the glycerol 3-phosphate dehydrogenase making FADH2. That's going to enter directly here near complex 2. Whether an electron enters at complex 1 or complex 2, ultimately both of these proton complexes result in the fully reduced form of coenzyme Q, shown here as the QH2. The QH2 is then going to keep going and continue to complex 3 and 4, as we will see as we keep going. All right, so now let's take a look at the individual complexes. For each of these, I'm pretty much going to stay on the picture while I'm describing it, but what I'm saying should be located on this slide, more or less. We're going to start with complex 1, known as the NADH ubiquinone oxidoreductase. Now that sounds like a horrific name, but if you take a look at it, it shows you what you start with, which is NADH here, and what you end up with, which is the ubiquinone, the fully reduced coenzyme Q. This is called a complex because it's actually 40 different proteins hanging out together, working together to accomplish a similar function. The way this is going to start, we need to be able to follow the electron flow. So the electron flow starts here as NADH donates 2 electrons to FMN, flavin mononucleotide. This is a cofactor bound to one of the proteins within complex 1. The flavin mononucleotide then continues to move the electrons through a series of iron-sulfur clusters, ultimately donating 2 electrons to this coenzyme Q to get the fully reduced form of the coenzyme Q. Now there's a few other things that are occurring as we look at this. We can see that we already are going to begin to move some protons. Because remember, according to the chemiosmotic theory, we're trying to move protons from the matrix, which is this side here, through the inner membrane to the intermembrane space. The first movement of protons we can observe is to just complete the reaction where we can fully reduce the coenzyme Q. Coenzyme Q needs not just 2 electrons to become this molecule, it also needs 2 protons. Protons are transported across the protein into the intermembrane space, actually 4 protons, as we complete the series of movements of electrons through the proteins. What we actually believe is happening, just to demonstrate this, is not just a big hole is opened up and protons are moving through. We actually see different amino acids undergoing protonation-deprotonation reactions throughout the proteins, shown here in this pink color, until the protons are moved to the outside area in the intermembrane space, and then they're released. Next we can look at complex 2. Complex 2 is known as succinate dehydrogenase. Again, this is the same reaction we've already seen. The enzyme is actually part of complex 2 in the electron-transport chain. Succinate dehydrogenase catalyzes the reaction in which succinate is oxidized to produce fumarate. During the oxidation of succinate, FAD is reduced to FADH2. So we have seen the initial movement of our electrons already. It's important to note, though, as we look at this reaction, that we do not see any protons being transported. So here's a look at what this complex looks like. It's going to have an area here for succinate binding, because succinate is the substrate of the enzymatic reaction. As the succinate is converted into fumarate, electrons are moved to FAD, then ultimately through iron-sulfur clusters, ending at a oxidized coenzyme Q, which becomes the fully reduced version of coenzyme Q. Again, no protons are moved through complex 2. Now we move to complex 3. Complex 3 is known as the ubiquinone cytochrome C oxidoreductase. Again, naming for the substrate and the product. This reaction is going to begin with electrons being donated from the ubiquinone or the coenzyme Q, and the ultimate product is going to be electrons being donated to cytochrome C. Now this one's going to be a little bit different because we have 2 electrons coming in from the coenzyme Q, but cytochrome C, remember, can only accept 1 electron. So this is a little bit more complicated. We have to explain how we can switch from a carrier that can carry 2 electrons to a carrier that can carry only 1 electron. And how we do that is called the Q cycle. But first, let's take a look at this picture and point out a few things. Complex 3 is actually 2 monomers, so you can kind of see how it's two different proteins hanging out together. In the center of this monomer is where we have the coenzyme Q binding site. The coenzyme Q is the substrate. That's what's coming from complex 1 or complex 2. Coenzyme Q is going to bind here, and then we're ultimately going to give electrons to this cytochrome C that's at the top. Again, we have some hemes and even a couple iron-sulfur clusters so we can move our electrons through. So now let's discuss this Q cycle. Again, I have it written in words for you, but I would rather look at some pictures to explain to you what's going on. When you first look at this, it looks very daunting. I agree. I remember being in your position, but I'm going to try to explain this to you in the most simplistic terms that I can. Remember what the substrate is. We're starting with coenzyme Q that is fully reduced and it has 2 electrons. So we start here. Coenzyme Q comes in. It's got 2 electrons. Well, we know we want to give an electron to cytochrome C. That's our product. But it can only take 1. So what happens is the other electron is transferred through a few hemes and into another molecule of coenzyme Q. But when coenzyme Q gets only 1 electron, it becomes this free radical version of coenzyme Q. So now move over and look at this side. Coenzyme Q with 1 radical will sit and wait. This is the cycle part. When a second molecule of coenzyme Q from either complex 1 or 2 comes in, it will do the same thing. Donate 1 electron to a molecule of cytochrome C, the product, and then another electron comes down, but this time there's a radical version of coenzyme Q waiting to accept the other electron. The second electron plus 2 protons pulled from the matrix produce another molecule of coenzyme Q. What do you think this molecule of coenzyme Q is going to do? It can pull back around through the membrane and donate electrons again. This cycle is allowing us to move electrons and protons at the same time. So let's break it down into the 2 different parts of the Q cycle and make sure we have a good understanding of what's going on and the overall purpose of each part of this. So again, step 1, completely reduced coenzyme Q from complex 1 or 2 comes into the binding site of complex 3. 1 electron is donated through an iron-sulfur cluster and a heme into cytochrome C. The other is pumped to another molecule of coenzyme Q. The moment this original coenzyme Q that was fully reduced gets rid of its electrons, it has protons on it too. Those protons go to the intermembrane space. So through cycle 1, we've accomplished 2 things. We've made a cytochrome C, meaning we've given an electron to cytochrome C to move on to complex 4, and we've moved some protons into the intermembrane space, which we were trying to do. Then in the second cycle, which my picture didn't come up, let's see, let's just go back here. In the second cycle here, we've got this radical waiting. Well, here comes another molecule of completely reduced coenzyme Q. It's going to do the same thing as it gives an electron to the cytochrome C. 2 more protons go into the intermembrane space. The secondary electron goes to the radical version of Q that we got from the first one, pulling in 2 more protons. So you can see we are continuing to donate electrons to cytochrome C, which we want to do to go to complex 4. And we are also pulling more and more protons from the matrix to go to the intermembrane space. The protons are pulled as they're moving into the radical form of the coenzyme Q, and they're being pumped into the intermembrane space as the fully reduced form of coenzyme Q donates its electrons. Cytochrome C, again, I apologize, my picture is not coming up for some reason as I'm recording this. Just wanted to point out again, Here's our cytochrome C that you can't see. But remember, that cytochrome is only capable of carrying that one electron. It got one electron from the end of the complex 3 that we were just looking at. And now that cytochrome C is going to carry one electron to complex 4. Let me try this. See if I can fix it. Nope, my images will not display. Well, we'll find a stopping point and then I'll restart so I can fix my images. Let's go ahead and talk about complex 4 so that we can find a good place to stop. Now that we're finally at complex 4, this is the point where the cytochrome C is going to bring another electron in and donate. The cytochrome C is going to donate the electron into copper ions that are located within complex 4. Once the electron is donated to the copper ions, oxygen is going to come in and take the electron from the copper ions. This is going to allow us to do a couple more important things. Well, we've had this electron that we've been jumping from one place to the next. The oxygen's got to be there to get and finally pick up these electrons. So if you're taking notes, you'd probably want to be on this slide, the electron flow through complex 4. What we're going to see is, again, cytochrome C is going to give the electron to the copper. The electron keeps moving through the copper ions. Then oxygen is going to move in, accept the electrons, and also pull protons from the matrix to ultimately produce water. This process, once oxygen is combined with the electrons and the protons making water, is going to allow us to pump 4 more protons out of the matrix into the intermembrane space.